Good morning, everyone. Let's go ahead and get started. My name is Jessica Vionis, and I am the Director of Marketing at Golden Helix. It is my distinct pleasure to introduce Gabe Rudy, who is our Vice President of Product Development. Gabe joined Golden Helix in 2002 and thrives in the dynamic and fast-changing field of bioinformatics and genetic analysis. He leads a killer team of computer scientists and statisticians in building powerful products and providing world-class support. Gabe says his passion is enabling Golden Helix's customers to accelerate their research. And you can follow Gabe on Twitter at GabeInformatics. And with that, I will pass it on over to him.
All right, thank you, Jessica. So what I'm going to be covering today in this webinar is some content that I actually prepared for a talk at the Molecular Tricon Conference in San Francisco just last week. Excuse my voice if I'm a little bit dry. Almost entirely predictably, whenever I travel, about 4 days later I get a cold, so that's just coming on here. But the content is going to be quite exciting overview of the question of how can one go about using public resources to try to get to a clinical grade analysis and interpretation of data. Before we get started on the housekeeping side, keep in mind that we will have some time at the end for questions, and feel free to use that little questions panel of your GoToMeeting interface. You can start asking questions now, and at the end, Jessica will help moderate as we get through as many of those as possible. And of course, like always, we'll be recording this webinar, and that will be available on our website, essentially be able to see it through YouTube. And I also am going to refer to the fact that we've done a number of these types of educational or just best practices or overview webinars, including ones about my own 23andMe exomes, which I'll touch on today. And I'm going to refer to those if you wanted to see some of the background or previous analysis that I did. Okay. So a little bit about me and kind of the type of perspective that I'll be lending to this question. Golden Helix, if you're not familiar with it, has been providing tools to researchers for just, for doing a genomic association and CNV and NGS analysis for all about 16 years. We're very proud of the fact that we're probably one of the few bioinformatic companies in our space that can say that we've spanned multiple millennium, and we really have had a chance to grow with both the community and the technology that has changed very dramatically, and especially with the introduction of next generation sequencing. And so my role at Golden Helix is with my wonderful team building products to enable this type of analysis. You'll note when I actually do some deep dives into some of these examples, I'll be using a visualization technology called Genome Browse. We do give that away as a free standalone tool. Go ahead and download it. We'll reference it at the end. And that's also the visualization engine inside our primary analysis tool, SNP and Variation Suite, which would have been used and will be used kind of for this type of work. So you can follow up on those, but that's essentially my background is the bioinformatic role of learning how to work with all these data sources and actually, very on a low level understanding all of their little gotchas, etc. So that's what I hope to apply in the context of where are we as a community and being able to, in a relatively straightforward manner, take the public resources out there and do a clinical grade analysis. So to cover that topic, I'm going to spend a little bit of time on what my background was in getting my exomes, as well as how to get that data that I have, which you'll see as a pretty raw data set, into a form where it's sort of appropriate to enter a clinical annotation and filtering pipeline. And then what that pipeline would look like to identify candidate interesting variants from a disease causing perspective, and then ultimately this comes down to our analysis is only as good as the annotations and public knowledge. Essentially, this is always the case in science. You build on the shoulder of giants, and what are the efforts for moving that forward going into the future to see how close we are to being able to be a widespread utility in a clinical setting. So how did I get my own exomes? Well, it actually is a very fun story, and I have a whole webcast and a couple blog posts that covers sort of it time frame by time frame. But in aggregate, what happened was 23andMe did, at a very limited time, offer a for a limited time, offer a pilot of they would sequence your exomes. I was already customers of 23andMe, and I realized it would immediately be more useful, both bioinformatically and from sort of an analysis perspective, to not just have my own narcissisome, but have my wife's exome and my son's. My wife, at the age of about 15, was diagnosed with juvenile idiopathic arthritis, and so it was an interesting phenotype, a complex autoimmune disorder, to see, to ask the question of whether an exome could shed light on that, and that I have had a previous talk where I covered that. But in this sense, in this case, actually, in this webinar, I'm going to simply look at mostly my own data from that raw exome sequence. And 23andMe was very clear. This was a pilot. It was experimental. They didn't guarantee anything in terms of interpretation or even the quality of data. But let's take a look at what that quality looks like. So this was, in all sort of objective senses, a high-quality exome. It covered the exon targets on an average of 105 X, so 105 reads covering those critical regions of the coding portions of your genome. And of course, it's an exon capture, so you're going to have that kind of pile of mountain effect around that coverage, and it drops off on each side. And you can kind of see the coverage here drops off to the tail. But all in all, it's a high-quality set of data to start with. And the question is, how do you sort of go about getting that to a point of being ready for a clinical analysis? Now, one interesting side note was I not only got one initial set of variants called by 23andMe, but 4 months later they decided they wanted to update some annotations, use a new bleeding edge version of GATK. Their alignment was done with BWA, and this is sort of a best practices pipeline circa 2012. And so their updated results they sent me about 4 months later, and I actually have a blog post about some of the initial analysis of that, which illuminated the fact that there was a bug in GATK that produced a bunch of false positives systematically in that second data set. But that's sort of a different story. What's interesting is also I went ahead and thought about, is there going to be a new way to analyze this raw data? And there was a company, still a company called Real-Time Genomics, and their whole mission in life was to produce a clinical grade secondary analysis, so alignment and variant calling. And in 2013, I had the pleasure of knowing their chief science officer, Francisco de la Vega, and he let me run my exomes through their pipeline. And one of the cool things about that analysis is that it took advantage of the pedigree structure. So when calling variants of my son, they were able to look at my wife's and mine variants at that site. So we could essentially improve the quality score, especially for things like de novo mutations, which it turns out he didn't have any after sort of having that high quality variant set inside at least the coding portions of genes. So the interesting thing then is I also thought, well, maybe there is a difference in sort of our best practices in 2014, and can we just look at taking a latest version of BWA? So I took the version released just a couple weeks ago, used the BWA MEM algorithm, which is optimized for reads greater than 70 base pairs, and I had 75 base pair paired end reads from 23andMe, and used a different variant caller, Freebase, which is a haplotype aware variant caller, and so to try to reflecting almost a best practices pipeline from today. Looking at the concordance here, if you take one of the more stringently filtered data sets like the 60,000 variants, you can see about 56,000 are actually concordant across all 4 different pipelines. But interestingly, there's quite a few that are called specifically by Real-Time Genomics and BWA Freebase, as well as plenty of room for variants that are called by a single pipeline. And you have to realize that, again, because this is exome capture, there are variants being called with very low read depth, and I did try to filter this down to reads that are greater than 10x read depth, but there's still a lot of room for saying what those filters will be to give you that high quality data set. And it's a heuristic based problem. Do you look at one score or another? What I used was Real-Time Genomics' variant set, and I used their sort of aggregate quality score based on recommendations from Dr. de la Vega to sort of set myself up with a high quality variant set. But it's actually, and this is a case I'd like to make, that it's not just about that single site and a single quality score and the heuristics that you can apply to that. So looking at this mutation, I'm going to try to make this case. So this mutation came up in my filters, and this is using the latest BWA Freebase pipeline, and it exists exactly as the same mutation. It looks exactly the same in the original 23andMe dataset, but it exists as a mutation one base pair after the donor splice site of PSPH. Now, it came up in my filters because PSPH is implicated in Mendelian disorders. Loss of function can result in pretty severe phenotypes. So, what's going on with this mutation that in that first two bases of a donor splice site, these are extremely highly conserved mutations in this region, almost inevitably result in a change in the splicing structure of that gene, and usually that results in a loss of function of that gene. So do I have a loss of function mutation? Certainly from a single locus perspective, this site looks very clean. Every variant caller would call this with very high confidence that I have a single deletion on one of my chromosomes. The purple here in this is a deletion notation. You can see it's about half of my reads, and that's what it looks like, a little gap in the sequence for the reads that support that deletion. But this is where the local context is vital to really, checking on and understanding what's going on. And the local context raised my eyebrows because, again, this is a pretty conserved gene. You can see our big population catalogs, 6,500 exomes from NHLBI, 1,000 exome or genomes from 1,000 genomes, have very few mutations here. Some in the splice site, this is a synonymous mutation, etc. And yet I have a big, high density, very, very close together set of mutations. So what's going on? Well, it turns out all those mutations are always on the same read. They're not being supported on both strands, so they're on the same haplotype, and the proportion of reads isn't quite 50%, so there's something going on. If you zoom out a little bit, you start to get a sense of what's going on. Both the 23andMe and BWA data has this pattern, and this is that exon. Here's that splice site zoomed out of reads, and again, these are always within the same read, very dense set of mutations. And essentially what's going on is those reads did not map in the real-time genomics data set or by the real-time genomics aligner because they had too many differences. BWA's default parameter allowed for those 5 mismatches and deletion and still align it as the best alignment. The alignment quality was actually, relatively high if you were filtering by alignment quality. It is the best alignment, but it's not that close of a match. It's not like there's only one mutation per read. So you can actually, and this is what I did, click on that read in Genome Browse, copy the sequence. Here's the 75 base pairs that includes those, covers right that exon junction, and go to BLAT, which is basically a way of taking a single read and aligning it to the genome on UCSC. So it essentially agrees with BWA. The position that it matches with 93% identity is exactly the same position that BWA aligned it to. There's a second match here, and that's what's interesting. You always got to kind of keep track of these second matches. Maybe there's a second place it should be. Should it be here? Well, it's another place on chromosome 7, and it has almost the exact same pattern of a deletion and mismatches. It's not in a clinical or a RefSeGene, high confidence gene. So maybe it's not that. Maybe that's all there is to the story. But still, this similarity score is not very high. It doesn't look like it's a perfect match. So let's broaden the search. So I went to GenBank and NCBI. You can take a sequence and blast it against any number of data sets in GenBank. And I went ahead and blasted this, or essentially looked to see if this matched any primary contigs, any primary chromosomes of any species. And it turns out it did. It matched 100%, exactly 100% of the length into the chimp and gorilla primary assemblies. And notice it's actually on chromosome 7. So I want to look into that a little bit more. So if I go and blat just the chimp genome, it matches chromosome 7 with that 100% identity, and it matches it almost in the exact same sort of positional location along chromosome 7. It's not inside of a gene, so it's not functional. So I have a piece of DNA. It doesn't, it looks like its best match in human is functional, its best match in chimp is not functional. It's a perfect match. So maybe that explains some things about me. I don't mind being more closely similar to chimp than gorilla in some places of my chromosome, but you know, I do kind of think that there's something else going on here, regardless of the similarities my wife might add as well. But the search being expanded sort of finds the missing piece of the puzzle here. So I expanded the search to not just primary chromosomes, but any human-based DNA in the GenBank archive. And sure enough, there's a 100% match to the scaffold of HuRef. So a scaffold is basically a piece of an island of DNA. In this case, it was about a 50 kilobase scaffold that was trying to be part of an assembly, but never got fully integrated into a chromosome with a positional placement. And HuRef is the J. Craig Venture genome that came out similarly in a parallel effort to the human genome sequencing effort. So essentially, Jay and I have this piece of DNA. It's actually his genome that's being assembled here that we don't have a matching place of the human reference. He has a scaffold that never got placed, but there's no mutation essentially. Our matches, this scaffold and the chimp indicates that there's essentially just a unplaced piece of human DNA not going into the primary assembly. And notice GRCH38, which is right here in this BLAST search, did not resolve this in the sense that this BLAST match is actually the exact same alignment that's on GRCH37 that we were looking at. In fact, if I pull up GenoBrowse and look at my GRCH38 alignment, it is exactly the same. So, let's talk about GRCH38 a little bit more. So in this case, it's an example of the local context discovering that there was a human reference genome-based issue where essentially a false positive in a variant call that looked functional was incorrect, not based on a variant, calling problem, but assembly problem. Will GRCH38 fix all of these issues? It certainly didn't fix this one, but GRCH38, it came out just in this last December, essentially 2 months ago, has, and I do want to give credit to the team, has really taking a step forward in terms of giving us a human reference to work with. A couple of different details on that. For example, GRCH37 never included a mitochondrial reference. So UCSC and 1000 Genomes used 2 different mitochondrial sequences. If you're aligning your data against one, you can't annotate against another. For example, GRCH38 resolved that by including the 1000 Genomes, the choice the 1000 Genomes had, the revised Cambridge reference sequence mitochondrial. So now we all have a common mitochondrial, but more importantly, there are a lot of assembly gaps, a lot of incorrect alleles fixed. Some of these gaps and fixes and patches essentially that are in GRCH38 cover functional places of the genome. There are new genes, there's all this good stuff, right? In total, if you look at my own exome, I aligned it to 37 and 38 with the exact same pipeline, called variants with the exact same tool, and I dropped about 20,000 variants. About the same proportion of all the different variant classes, but there's 20,000 fewer variants, which essentially should probably mean 20,000 fewer false positives. And in fact, if you use the transition to transversion ratio as a metric of false positives, you can see it's actually gone up, which is great. For exome capture, biologically, you might expect this a true number of, with 100% true positive variants to be about 2.5 or 3. So the fact that it won out by 0.05 does indicate that all in all, the data that I have aligned and called to GRCH38 has fewer systematic alignment, variant calling, or sequencing artifacts, or those actually alignment or variant calling artifacts. But I actually argue that this in a little bit more detail in a blog post I just put out on Monday, that we probably won't be using GRCH38 for a long time. It's a chicken and egg problem, obviously. Our annotations aren't mapped to 38, they're mapped to 37, and the gap between sort of, when the data starts to arrive in 38 from an annotation and consortium's perspective and when it's useful to you, I think is going to be a bit more painful and larger than between our last transition of NCBI 36 to GRCH 38. So if you can go ahead and see more details about that in my blog post, if you like, feel free to comment if you disagree with me. Be curious to hear your thoughts. So anyway, I talked a little about getting to a high quality variant set. What about the analysis portion? And the analysis portion is heavily dependent on annotation, as well as what are your choices on how you get to a high quality or a candidate set of variants. So those choices are obviously a very subjective thing. We've done a number of different types of tutorials and workflows where we've worked with customers and defined their best practice for doing this type of annotation. But I think from a clinical perspective, you have to probably refer to this landmark paper that Baylor College of Medicine just published right before ASHG last year, in which they went through their own sort of case history of going through their first 200 exomes that they ran through a clinical setting and got to a final sign out on. And this is the diagram from that paper of their workflow. Now, obviously, each of these boxes are referencing, data sets, and we'll have to talk about how we can kind of replicate this workflow. But to give you a high-level overview of what the strategy is, they essentially break things down into a focused report and an extended report, where the focus report is something you definitely as a physician will be looking at, and if you want to get down into the weeds a little bit, you can get into the extended report. And the first box of steps here is really about finding variants that are in disease genes associated with the phenotype. So these are essentially the first cut of variants you want to look at, implicated in a gene that's related to the phenotype, and high quality variants that are likely to be damaging to those genes. The second set of data is if it's not in the disease gene related to the phenotype, maybe it's in a medically actionable gene, we really should report it because there might be something you can do about that information. And similarly, if you have a carrier, if you have a mutation in a gene that is one of these carrier status genes, it would be helpful to pass that on potentially to the patient so they can use that information in their own planning. Once you get into the extended report, you get into this list of deleterious mutations that are not in any of those specific set of genes, as well as variants of unknown significance that are potentially damaging to a gene implicated in Mendelian disorder, but that gene isn't already been known to be associated with a specific phenotype that you're looking for that case. And then you get this sort of bucket of deleterious variants and genes that who knows if they're implicated in any sort of disorder. Okay, so I'm going to try to replicate this on my own exome. So what do? Well, first I need to get all of these annotation sources. And they were not specific, and when they said filter by 1,000 genomes, they just said filter by 1,000 genomes. That's horrible from a reproducibility perspective, but let's all just assume that they are using the latest version of all of these data sources. So for 1,000 genomes, we're using phase 1, which is their final sort of variant set of their 1,092 samples. And for they reference the ESP project, so we're going to use that National Heart, Lung, and Blood Institute catalog of mutations across 6,500 exomes. They have a V2 release of that. Very important to notice the distinction between their different release numbers. They also filter on whether something's an HGMD. Now this can be a bit trickier for me to follow. HGMD basically has a public and a professional version. I'm sure they're using the professional version. The public version is only free to academics. But Ensembl has a variant catalog on their sort of database, which they simply flag the variants that they know are in HGMD public. So I can use that catalog as a way of knowing whether something's in HGMD as an approximation. Finally, you can annotate about whether a variant affects a coding region of a gene or its potential splice sites, like that one we were just looking at in my genome. And then they get into this list of filters that is basically, which genes do you prioritize? Well, one, you prioritize genes in single gene disorders. To approximate that, and I'm not sure this is these start to be more subjective, but I'm using OMIM where any inheritance status has been classified. So essentially, you have a Mendelian model for that gene, and it's an OMIM, so it's likely it is associated with a Mendelian disorder. For medically actionable, they probably have their own list here, which they didn't divulge. I used this set of 114 genes by a study published by NHLBI, where they looked at medically actionable variants in 1,000 of this cohort of 6,500 individuals. A great paper, and they also listed all 114 genes that they deemed medically actionable. For which genes are dominantly and recessively inherited, this is actually a little trickier than you might think at first. We tried a couple different ways of getting to this information. And then finally, I also just asked Twitter, does anybody know the best way to define this? And I got a response from Deanna Church, previously from NCBI's Genome Reference Consortium, and she mentioned the fact that MedGen is an NCBI resource that catalogs a lot of clinical annotations of genes, including the known inheritance model has been divulged by researcher clinical data. And finally, that carrier panel information, you can pull up the ACMG incidental findings guidelines. There's a separate sort of more vague guideline about which genes are carrier panels, and so we try to use a combination of those things. So here it goes. I'm going to do the analysis in PowerPoint form because it would be a little more laborious to do it interactively in SVS, but this was all done in our analysis software. And the first step is just essentially, getting to high, rare, potentially damaging variants. So they say if you're in HGMD, go straight, past go, collect $200, and then you also want to just make sure it's relatively rare, so less than 5%. But if you're not in HGMD, go through a little more selective filtering, that you're in a protein coding region or you have an extremely rare, less than 1% allele frequency. So following that logic, I get about 840 variants. And they think about 400 to 700 variants on average end up being in genes associated with single gene disorders. And that's actually about right where I was. 644 ended up being in genes associated with single gene disorders in my exomes. I'm skipping the whole block related to genes associated with a phenotype, since I have no dramatic phenotype to report. And so the next step is to look for various variants that are deleterious. And so of these 644, 20 are expected to be loss of function. These are frame shift, nonsense mutations, etc. But none of my 20 are medically actionable or in carrier genes. Fine by me. I did dig into these 20 in a little more detail, and again, this is a high-quality variant set, and I would, I think I can spot about 1 or 2 false positives, but all in all, I have about 20 loss of function mutations, potential putative loss of function mutations in Mendelian-associated genes. What does that mean? Well, it doesn't mean much, because a lot of these genes are haplosufficient, and I just have essentially one potential knocked out copy of those genes and another functional copy. Interesting, though, from a carrier perspective, potentially. So of these 624 that weren't of these loss of function category, you can look at if any of them are in a disease with a dominant inheritance, right? So then in a heterozygous state, they were potentially extremely relevant. I don't have any of those. What about those that are in a hemizygous state on the X chromosome? For me, I'm a male, or are potentially this 2-hit model where you knock out 2 copies of a gene, or I'm in a homozygous state. That's a bit trickier to come up with that for myself, because this is a single N of 1 study where I don't have my parents' sequence, so I don't know which strand or which, if I'm looking at a paternal or maternal heterozygous variant to classify a compound heterozygous. So that gives essentially a giant bolus of data in this extended report, nothing in my focused report. A lot of fun to follow that. And again, the point here wasn't to essentially go through each individual mutation, but really to kind of highlight some of these challenges of getting the data and doing the annotation itself. So in that context, I actually want to look at a mutation that was one that I touched on in a previous webcast, because it was listed as pathogenic in dbSNP. And essentially the state of data in the community has changed And now the story about this mutation has changed a little bit. And that's a good thing, right? We expect that hopefully data sources change in the public. And so I'm going to walk you through, though, what that looks like for my mutation. So I'm in the OTC gene, a gene implicated in a pretty severe metabolic disorder. If I zoom in into this exon, you can see my exome sequencing here. Very nice coverage across this exon. And this allele that I have, I'm in an X chromosome, by the way, so I should expect to be hemizygous for anything that I'm looking at. This allele is supported very heavily on the forward, reverse strand, etc. And essentially, as a no-brainer, I have a G here or an A here as opposed to the reference of a G. So if you look in dbSNP at this site, there is cataloged 3 different alleles. That's why we have the little tricolor box, this AGT. If I click on that, I can see information from dbSNP. So let's go ahead and look at what I originally saw when I popped open dbSNP when doing my analysis, and that is this this mutation. Has the by looking at the RSID for this mutation, it says I have a pathogenic allele, and that's essentially the depth of information I had a year or so ago. But remember, we do have 3 known alleles present at this site. If I look at this VarView channel, it is now much more detailed than it was a couple years ago, and I can look at the fact that the G to A mutation is reported as untested, and the G to T mutation is the one reported as pathogenic and implemented by 3 different labs, 3 different sources essentially. One as a metabolism a disorder test, and one just from OMIM. You can also pull up this mutation in Clin Var, which I'll touch on a little bit later, but this mutation has been submitted again by OMIM. You can see which alleles are present, and going back to my data here, so this my allele was a G to A mutation, right, not a G to T, and that one was not implicated as being pathogenic. So essentially, just by matching on RSID or by position, it's not the whole story. You have to actually do the proper allelic-based annotation to know that, while I may have a mutation here, it is a missense mutation. I think it actually results in a different amino acid than the G to T mutation that was pathogenic and does have a known clinical implication. So just an example of this, this need to go beyond sort of the maybe more obvious approach of just finding which RSID is associated. The next question then is I was able to show that it's possible to follow a workflow to get to a list of candidate variants. What about these public resources? And as I mentioned, dbSNP actually has a much better sort of view of these reported pathogenic alleles now than they did a couple years ago. But there's a number of different contexts in which public resources are focused. And so if you're looking at germline mutations, you're going to find, dbSNP is a very expansive catalog. It actually has both somatic and germline mutations in it. And it essentially will take any mutation anybody reports. So it's not like it's a useful way to say whether something is not a false positive or whatever. But there's other resources for defining germline clinical status, and I'll touch on those a little bit in a second. And those are relevant to both carrier screening things as well as the types of diagnostic odyssey searches like Baylor was doing with their exomes. There's also going to be resources for predicting lifetime risk. So this can be lifetime risk of developing cancer or, type 2 diabetes or Alzheimer's. I would say in a general sort of covering all lifetime risks, 23andMe's probably got the best platform for doing that, and I think it's just a matter of time. I was talking to their chief science officer at TriCon a little bit about this. Obviously, they're heads down working on appeasing the FDA's request for information. They have these big, 1,000-page documents, so they will be, in this business of predicting lifetime risk. But there's also the more focused approach, predicting lifetime risk for cancer, etc. And we'll touch on that a little bit too. I won't go into much in the way of oncology care or PGX. The data sharing problem is very different because when looking at, say, somatic mutations, you need to essentially have all somatic mutations for a sample shared to have sort of a meaningful aggregation of information about that sample. Unlike the potential ability to share, say, my single OTC variant and a little bit about myself and be able to potentially interpret it with just a single mutation and information. Pharmacogenomics has also got a different sort of landscape of data sharing because there are actors like testing labs and pharmaceutical companies who have a lot of different interests. There are some work to create sort of a pre-competitive data sharing spaces specific to pharmacogenomics, and there's discussions going on at different levels, both government and industry, to try to make those types of data sharing resources available. On the germline side, I would say the biggest actor and game changer in this space has been Clin Var. So I talked to Heidi Rehm, one of the founders of Clin Var, who also runs her own diagnostic lab, very similar to how Baylor is doing those exome diagnostics. She does exome diagnostics at Partners Healthcare, and in that context, she really felt the need to be able to take cases and their decided pathogenic status of individual variants and share them. And even though that sounds like something that you're giving away a competitive advantage, these data sets aren't going into published literature because they're clinical. And without a data sharing, overall the patients lose because these are such rare disorders that unless data sharing is happening at a national and global level, we're just simply not going to improve our success rate. And all in all, we all, there's going to be this essentially benefit from both a individual labs and a patient perspective if there is a current place for all this data to be aggregated. And so that's the mission of Clin Var. It's really not a type of resource that is very selective in the way, once you say that your clinical lab and validate it, you should be able to get your data submitted. So it's not cleaning up or, say, having an expert panel review each submitted mutation. It has sort of an orthogonal way of ranking variants, essentially believability or trustworthiness, by giving them a 1 to 4 star rating. And at the level of 4 stars, there's a requirement of like a disease-specific committee or consortium agreeing with publications and etc. That this is a highly penetrant mutation that will have a very direct and measurable response on a phenotype. If you're at that level, you can be pretty darn sure that you're looking at high-quality annotations. So there's a big effort to essentially have all labs be submitting their data to Clin Var. It's just getting going. It's a couple years old, but already there's a number of labs that have submitted. These are the top 5 submitters: Emory Genetics, Samuels, and Vitae, which is a private company, OMIM, etc. So another data source that was used in the exome paper, as well as probably most clinical labs have a professional license to this, is HGMD. It's been around longer. It has a primary source of data coming from research literature, so they've manually curated every time a paper says, in this specific disorder, we think that this is the causal driver mutation. They similarly have these pathogenic statuses of whether something is labeled as pathogenic, probably pathogenic, variant of unknown significance, likely benign, etc. And the difference here is, though, but all of these things require manually intensive work to maintain these databases. HGMD's model is to support that work through having a professional version that is a paid-for license. So if you're a lab, you can buy a yearly license. There's different levels of access to it. They have a public version. You can log in as an academic to see that. It lags behind in terms of the content and detail, but it is a place where you can pull up some access, sort of a hunt and peck way, as opposed to having the pro version where you can actually annotate your data in mass against HGMD. So an example then, let's apply again this information about Clin Var and the way clinical data is aggregated to a very high-profile carrier screening case, and that is testing for pathogenic carrier mutations in CFTR. CFTR is one of these cut and dry places where if you have some of the more prominent pathogenic alleles and you have two of them, you essentially have two loss of function copies of CFTR, you will, essentially with dead certainty be diagnosed with cystic fibrosis. And it's been tested for a very long time because 1 in 30 Caucasians, for example, are actually carriers of delta 508, one of those highly confident pathogenic alleles. We'll touch on that in a second. But on a database perspective, it's interesting to watch its evolution. Again, it's been tested over a number of years. CFTR2 is a database specific to cataloging clinical alleles in CFTR, curated by Hopkins. It's a version 2 database. It itself was an aggregation of 3 or 4 previous generation databases. ACMG's got its own list, etc. But now you can pretty much find all of those alleles from all of those databases in Clin Var. So Clin Var's got over 1,600 variants annotated across CFTR's gene. About 400 of those are marked as pathogenic. ClinVite, which is another clinical database, also has about 400 variants, 300 of those marked as pathogenic in CFTR. So what's the caution I'm noting here in terms of annotating this? And this is actually a great story. So delta 508, like I mentioned, is one of these high confident alleles. In fact, it's one of the few variants in Clin Var that has that 4-star rating I mentioned. And very, very few actually. I probably should have pulled up the number. So it's essentially a 3-base deletion along the coding sequence of CFTR that results in the 508th amino acid residue, this phenylalanine, being deleted. And it's sort of canonically called delta 508, but you can call it, various different things if you want to. So let's go look at what this mutation looks like from both a database and a next generation sequencing perspective. So going back to my little list of projects here, I'm going to pull up. So again, I'm using SVS because SVS's 8 version incorporates Genome Browse as a technology, but this view here, I'm looking purely at public databases and BAM files and VCF files. This view you can get to by just having a version of Genome Browse standalone, which is a standalone free tool that can visualize all this stuff. So I'm looking at CFTR here. You can see it's a very long gene. If you remember that little protein diagram, it's got a number of different domains. But I have some data here which was designed to test this gene in a carrier screening panel. So it's actually amplicon data. And if I zoom on into this this exon here, exon 11, you can see the amplicon depth at different levels. And if we zoom on in to the place where you can see where that 508th phenylalanine is, this is the 508 that gets deleted in a delta 508. In this case, it's present in the NGS data in a nice, clean, heterozygous state where half of the reads support a 3-base deletion. Looks all fine and dandy. Here's that CFTR2 database from Johns Hopkins. It's got that delta 508 annotated right here. Here's 2 different versions of Clin Var. It's also got that delta 508 annotated. And here is dbSNP 138. Okay, so what's going on though is it gets a little more tricky than just describing a single mutation. The reason is describing this mutation is ambiguous. Why is it ambiguous? Because this deletion happens to span this haplotype. If you read the reference here, it's CA, TC, TTT, so 3 T's and a G, okay? And what matters is you lose 2 T's and a C. So I can describe that as this TCT here, but if I just started the deletion at here, this position, and described it as a CTT, you also lose the 2 T's and a C, and the haplotype is the same. Now, it's all about sort of standardizing, right? It's fine if something's ambiguous as long as you say, okay, we're always going to describe it this way. The problem is there's 2 different standards. One is the way we describe things when we do NGS analysis. We always prefer to describe things in their leftmost position according to the reference sequence. The reason being the reference sequence is the only thing that we can sort of refer to across the entire genome. We may not be in a gene. We have to use something like the reference sequence. So the leftmost position is the description of, as it is here, the deletion of a TCT. When we describe things in terms of genes, we like to describe them in terms of their more functional elements, like HGVS notation. And in fact, the canonical way of describing delta 508 is that second position of the CTT. And here it is in dbSNP 138, that CTT deletion. And if I click on that RSID, sure enough, it will come up in dbSNP 138 with that pathogenic allele status. And in fact, you can pull it all up in Clin Var, like I had that picture. This is how it's described in Clin Var. It is a deletion of a CTT here with a pathogenic allele. That's fine, but the problem is if you were to annotate this this NGS data against this CFTR database or dbSNP, you would not hit that pathogenic allele if you're just annotating by matching on sort of the chromosomal position and alternates. Now the story goes on a little bit more. So if you notice, dbSNP actually does have a record where the TCT is deleted. Remember, these are exactly the same DNA mutation, just being described in two different genomic reference alternate positions. This dbSNP record was submitted by the 1,000 Genomes source. It turns out of the 1,000 genomes, about 12 were present, had that delta F508 mutation. So again, dbSNP pretty much takes data from any source, and 1,000 Genomes uses an NGS-based pipeline, whole genome sequencing, called this variant in 12 samples, called it using the canonical description, submitted it to dbSNP, and now we have two different duplicate entries with two different, for the same mutation with two different annotations. So that's what's tricky about it, and you can see Clin Var also faces this issue. Here's the latest version of Clin Var. It only describes the delta 508 at this position here. So the solution here is that you can actually do one of two things. Whenever you're going to import your data for analysis, you can always move it into a canonical left-aligned form, or you have to actually change your annotation mechanism so it can account for multiple representations. And this is one of the more tricky ones where you have multiple alleles. If you have like just a homopolymer of like 3 T's and you have an insertion of another T, it's pretty obvious that an insertion of a T here versus an insertion of a T there result in the same thing. But it's less obvious sometimes, like with this mutation. But the problem is this mutation is critically important to get right when you annotate it, because it is the most common, good old 80% of cystic fibrosis cases have CFTR delta 508. So it's very important from a clinical perspective to get this one right. Okay. One more story to go through, and then we will take some questions here. And that is the story of annotating that lifetime risk of BRCA1 or breast cancer through the BRCA1 and 2 gene. Of course, if you're in this space, you'll probably be familiar with the fact that Myriad Genetics has had a patent on the testing of BRCA1 and 2 since 1995. That patent guarantees them a monopoly on the ability to run genetic tests, and that has provided them through the hundreds of thousands, if not millions of tests they've run, with a database that contains very, very detailed and relevant information about which mutations are pathogenic, etc. Well, the world changed in June 2013 when that patent was overruled, and now, any lab with the know-how to set up a test can actually run and provide reports to patients based on their BRCA1 and 2 mutations. The problem is the public annotations databases are a good decade behind what Myriad has for annotating which variants are pathogenic. We simply do not have that bolus of data since it's been monopolized by Myriad. So there was sort of a clever solution to this. The folks at Clin Var as well as Invitae have their own initiatives. Invitae's is called Free the Data, which essentially gets you a back door into Myriad's database. So if you were a patient which had a BRCA1 and 2 test run by Myriad, you would have received a piece of paper that says, here are your mutations. We classify this one as pathogenic, this one is not benign, etc. And these initiatives are essentially telling you, go ahead and email us that report or scan it or take a picture on your phone. You'll win a free iPod. So they're encouraging that. And through that initiative, many people people have, and ClinVar itself has this source called the Sharing Clinical Reports Project that has about 2,000 mutations. These are essentially myriad genetic mutations and their myriad genetic classification being added to ClinVar. And so now that data can be publicly used to look at things. I'm not going to touch on this too much, but ClinVite is Invitae's release of their own curation of pathogenic, and likely pathogenic, benign, etc., clinically relevant mutations across about 200 genes. They used ClinVar as well as a number of other labs that have shared their data. Most of this data will always will end up in ClinVar eventually. I was talking to Heidi Rehm, and she doesn't necessarily encourage using ClinVite, and she doesn't want us to divide effort. But at the moment, it does have a number of more mutations annotated than ClinVar. The trick is it's annotated only in terms of their coating-protein changes. So they're annotated in terms of their HGVS notation, and not all of those were able to get genomic mappings when we try to essentially map ClinVite to HG19 or GRCH37. But we got quite a few of those to map, so let's take a look at what those look like in the context of my wife's BRCA1 and 2 genes. Could look at mine, but I thought it'd be more interesting to look at my wife's. But spoiler alert, nothing interesting to look at, but it's still fun to look at the annotations. So here's BRCA1, here's the exome coverage from 23andMe, and here's ClinVar, 2 different releases, and ClinVite. And if you zoom on in here, you can start to see we have a lot of mutations covering these genes, sometimes more mutations in the new release of ClinVar, sometimes less. The ClinVite annotations has a number of different fields here, and I can label, this based on one of these fields, like the reported classification or the inferred classification, etc. And you can see that some of these are annotated as being pathogenic in these BRCA1. But my wife has no BRCA1 mutations. Let's go ahead and look at BRCA2. Looks like she does have mutations here along the 13th chromosome. And so we can start to look at these, and I'll pull up the table version of my wife's data. And if we zoom on in, okay, I'm in the UTR of BRCA2. She has a heterozygous A. I can look at the alignment data. It looks nice and clean, but ClinVite annotates that as benign. So I can go ahead and jump through and look at a number of different variants. This one's in an intron. She has another intronic variant, but they're all annotated as benign. Notice ClinVar does not necessarily annotate benign variants. They only annotate pathogenic, so there's more here, information about which ones are not to be that interesting. Here's one that's not in a database, but it's inside an intron, etc. So another one inside of a coding region, so I'm in the middle of this exon here, but again, her C allele is annotated as benign, etc. And then one final thing, I'll stop on this. We're in the middle of an intron, and I have a clean mutation. The mutation, again, is in one of those regions. It's an AATTAA sequence. You can describe that in a number of ways. You can describe it as a TTAA, an ATTA, a number of different ways of deleting 4 bases and still describing the exact same mutation. And again, ClinVite has got another description here of this as being a variant of unknown significance. So just another example of annotations being a bit tricky when it comes to these insertions and deletions. Okay, so with that, I do want to, didn't want to do that. I wanted to finish up with one last section here. And that is essentially, so we have ClinVar. Are we there in terms of being able to set up labs that do this type of work? And it's actually, it really depends on what type of work you're doing. Like that carrier screening stuff, it does look like you can, for the most part, find all of those interesting or useful annotations and set up a carrier screening panel and not have too many difficulties. But if you're doing what Baylor was doing and going on these diagnostic odysseys, it turns out, and again, talking to Heidi Rehm about what her experience was, is that most of the time you find 1 or 2 variants that are likely to be implicated for the patient, but they're almost always rare or extremely rare or novel, in which case then you need this big set of skills, so this cross-training set of skills to do that interpretation of, is this novel mutation likely to be, one that's driving this phenotype? And her experience was, just hiring MDs or pathologists, they didn't really have a sufficient molecular genetics background. She really needed to be hiring molecular geneticists who had enough of a background in the clinic and at sort of a bioinformatic level to be able to interpret this data. And there is a certification that sort of covers this, and that would be the Clinical Molecular Genetic Certification by the American Board of Medical Geneticists. But when they started that certification in 1995, since they've started that certification in the mid-90s, they've only had 500 people pass that board, and who knows how many of those are actually actively in practice. So this certainly isn't a common or easy skill set to acquire. I really appreciate Baylor's effort to share sort of their own thought process. They have this clinical sign-out conference where they have YouTube videos where they go through their own, they usually have one of the MDs or clinical geneticists talk through their own thought process and getting to the sign-out report, describe the novel mutation in the context of the gene, the phenotype, the system biology, etc. So, that kind of sharing is really going to accelerate this and make it so that we can have more of a possibility of doing this type of work in larger scale if that's something that's required. I do want to thank all the people I was able to talk to in preparing for this content, especially Heidi Ream, and also a number of people, one from Biobase and Invitae as well. And finally, at Golden Helix, we're gearing up for our next major release. Greta Lindsay Peterson is our quality manager and director of SVS's product management, but she took some time to help me with this analysis, and I really, really appreciate that. We've all been really busy here. So with that, we will take some questions, and as a reminder, we will have this recorded, but let's go ahead and stay on the line for a while and hear if anyone has some things to follow up on here.
Thanks, Gabe. Your webcasts are always so interesting. I got the chance to listen to a dry run this morning and then again just now, and I pick up something different or new or interesting every time, so thank you so much. Feel free, if you haven't already typed your questions into the questions pane, you can go ahead and do that now. We do have a couple in there. And I did want to mention that if you haven't already downloaded Golden Helix's free genome browser, which Gabe showcased using the showcast in the examples during the webcast, you can download it at genomebrowse.com, and we do have builds for Windows, Mac, or Linux, so you have some options there. Gabe also talked a little bit about running his exomes through the Baylor workflow using Golden Helix's data analysis software SNP and Variation Suite. If you would like to have a trial of SVS, please do visit our website and fill out an evaluation request, and we will get right back to you on that. Gabe mentioned this, but we did go ahead and record this webcast, and we'll be putting the recording as well as the slides up on our website hopefully by tomorrow. All right, let's go on over to some questions. The first one we have is from a customer, actually. Since I'm already using SVS, are there any situations where I would need to use your genome browser tool on its own outside of SVS?
It's really a matter of preference. I always have SVS and Genome Browse installed as two separate applications, and sometimes, I can double-click on a BAM file and quickly look at it in standalone Genome Browser. But more and more, I'm doing most of my work in SVS, and I like being able to kind of have multiple windows, like I did with this project, sort of side by. Oops, that's my Emacs editor. Have multiple sort of views of data, as well as actually having some analysis side by side and saved in a project context. So that kind of thing is nice about the integration. It's really a matter of preference, and I still suggest having both installed. They will share a lot of the same data. If you downloaded data in one place, it'll be used in both contexts.
Okay, great, thank you. for the TSTV calculations, it looked like one was 2.0 and one was 2.1. Are those results really, or those differences really significant?
You know, it kind of have to understand the range of TSTV. At a whole genome scale, it may only ever approach to at even a high quality data set. Of course, I'm looking at exomes, and so it should be enriched. A TSTV ratio of high quality variants inside coding regions is more like 3. So just even that little bump, it does indicate that, maybe surprisingly, changing the reference sequence actually just reduced false positives without doing any work. And that's the total set of variants without doing a single filter on those two sets. If I were to look at TSTVs between, say, high read depth or high confidence variants, it'd be maybe another interesting way to approach it. But in aggregate, I thought it was interesting that it went up at all, and it went up enough to be noticeable.
Okay, great, thank you. We had actually several people ask if you had the reference for that Baylor paper.
Yes, I will, let's see, I think what we'll do is just append that to these slides. It's really easy to search for. If you just look for clinical exome sequencing Baylor, it's probably the first thing in Google. In fact, let's just do it, clinical exome sequencing Baylor. So it's this one, whole exome sequence, or this whole exome sequencing Baylor College of Medicine. It's published originally in the New England Journal of Medicine. There's a couple of different references to it. Oh, nope, that's not it. That's their advertisement. So here it is. Clinical whole exome sequencing for the diagnostic of Mendelian disorders. Yes, that's it. So they went ahead and sequenced these 250 probands. I'll leave that up if anyone wants to write it down.
Okay, thank you for that, since we had several people who were asking about that. Is there a way to extract only the healthy samples from the ESP database?
Yeah, so that's an interesting question because really there are no clear definitions of healthy samples. In fact, ESP as a cohort is explicitly not healthy. It is being done by the National Heart, Lung, and Blood Institute, and if you look on their website on the different groups that are in that total set of 6,500 individuals, they all have extreme phenotypes in one manner or another. Some have extremely high blood pressure, for example. So even the 1,000 Genomes Project, we have no strong phenotypic annotations for those 1,092 individuals in the phase 1 group, and now it's going to go out to about 5,000 or so individuals in the phase 3 group we'll see in the next year or so. So at the end of the day, we're just sort of hoping that in aggregate population catalogs capture a slice of sort of allele frequencies across humans. Many of these groups, including Baylor, have their own internal control databases. And that makes a lot of sense too, not just from a perspective of you have known healthy people in those controls, but if you're running an NGS pipeline, you are going to have technical artifacts that are entirely specific to your exome capture, your pipeline, your bioinformatic tools, and you'll find that there's variants that are not in the public databases, but turn out to be in 30% of your controls. And so they're very easy to sort of weed out of your analysis by having your own internal control database.
Okay, well, I think we'll go ahead and end it there. Thank you again so much, Gabe, for giving that webcast again today. And I hope that you all have a great day.
All right, thank you guys.